Alright, well, hello. This is the first video I make for the Butler County Community College students. And as we agreed, the main purpose of this video is to cover aspects that we may not have time to cover at the lecture in an expanded way. So I will make this video and subsequent videos as well to help you with your reviewing of those chapters as we go through them. So we did cover quite a bit when we talked about the DNA and the function of the DNA. We talked about the genes, the genome, which is the entire genes of one person. We did talk about the chromatin, and we discussed that the chromatin would be composed of DNA and the protein that is used to be wrapped on. We said that this protein is called histones, and they're very, very important players of gene regulation or regulation of gene functions. The nucleosome will consist of the core particle that is consistent of histone and DNA. Then we have the linker DNA that connects each particle with the subsequent one. The chromosome is a very long piece of DNA that is wrapped around the nucleus, that is wrapped around the histones and packed very well into certain structures we call the chromosomes. On the chromosomes, you will see certain genes have a specific location. So let's say that the gene that is responsible for your insulin will be kept on chromosome, let's say 5. But not just anywhere on chromosome 5, it has to occupy a specific region within chromosome 5. And the same thing for any other gene, so everyone has its own address. Every gene has its own address on the chromosome. And that is very, very important. Any change in the position or the orientation of that gene, of course, will affect that gene function. The typical or classical image of a chromosome we think of is this X-shaped structure, but that really happens only when the cells are dividing, because what you're looking at here is a duplicated chromosome, or what we call two sister chromatids, and they're connected together by what we call a centromere in the middle. This kind of shape we only see during cell division, active cell division, because such packaging into a definitive shape for the chromosomes only happens right before cell division. RNA will differ slightly from the DNA. It still contains base, sugar, and phosphate, but the sugar here is different. It's a ribose instead of the oxyribose. And the base, we still have the C, the G, but instead of the thymine we will have uracil. So uracil is the only difference when we come to the nitrogenous bases between the two nucleic acids, the RNA and the DNA. On the other hand, the RNA is a single strand structure, whereas the DNA, as we know, it was double-stranded. As we said earlier, the genome is the collection of all those genes in your body, and when they started to do what we call the Human Genome Project, we thought that we would find hundreds of thousands of genes, but we ended up with about 25,000 genes or so. The thing about it is, as we will talk about later, and we did cover that in the lecture, is the alternative splicing, the ability of a cell to produce multiple proteins from the same code, from the same gene. And we will see how that takes place. That leads us to what we call the genetic code. The genetic code is a system that enables the 4 nucleotides, the A, the T, the C, and the G, to form amino acids. So if we pick 3 of those, and it can be AAA or TTT or ATA or so on, This kind of combination and the alternating those letters with each other or with themselves will give us different codes, and then we can have specific codes that will encode for about 20 amino acids. Of course, we will have many more codes than the amino acids, because if you think of those 4 letters, if you make the combination, you will end up with many more possibilities. And so many amino acids will be coded by different sequences. So it will be alternative sequences, and still it will encode for the same amino acid. We also have specific codes for stopping the translation, and we have specific codes for initiating the translation. So not every single code will encode for an amino acid indeed. This is what I mentioned, the base triplets, and they are a sequence of 3 DNA nucleotides. We can have 64 possible codons, but we only have 20 amino acids. So the 61 that code for amino acids, we can have multiple codes for the same amino acid. Then we have, in addition to those, we have stop codons and start codons. The stop codons like UAG and UGA and UAA, those would be the stop codons for the translation, whereas the start codons like AUG would encode for methionine, and that will begin the amino acid sequences, and that will begin the process of building up the amino acids to make proteins. In terms of protein synthesis, we did cover that aspect in the lecture, and we agreed that the DNA will hold our recipe. Then if we want to synthesize a protein out of it, then we make a copy of that recipe, which we called messenger RNA. Now the messenger RNA will be translated by the ribosome and will start building on the amino acids that are carried by transfer RNA to form to us the proteins. So there are 2 processes. One would be called transcription, and this is essentially copying the DNA into RNA. How do we do that? We create bubbles within the DNA, and then an enzyme called RNA polymerase will bind to the DNA and will start assembling the messenger RNA. We need to remember that the RNA does not have thymine in it, and it will be replaced, on the other hand, by uracil. The RNA polymerase will build up those this structure, and we will call that pre-messenger RNA, and we'll talk about the pre-messenger RNA in a short bit. And that is not the final product. Let's talk about it now. As we said in the lecture, that will represent much more than is actually needed for making the proteins, because it contains some what we call nonsense or junk portions of the genome that is not needed, and they must be removed in a process we call splicing of the RNA. The exons, on the other hand, they will contribute to making the codes that will encode for proteins, and those will be translated. So from the pre-mRNA, we have area that makes sense and will be translatable, and there is another area that will not be translated, and therefore we need to remove it. Again, if we look here is the DNA, and it will encode for pre-mRNA, which is very long, as you can see here. And those areas here, they're much longer than that. Actually, it's much longer than that. These are fairly short, just to give you a demonstration. And those don't encode for any protein, so they have to be spliced out before the RNA is actually processed. And when they are spliced out, that gives you lots of possibilities. A can be with B and C, and that gives you one possibility, or A with B and C and D and E and F would be another possibility, or B and D and E, that's a third possibility, and so on. And we call that alternative splicing, and in this way, one gene can make more than one protein, and it's a very interesting and important aspect through which the cells can make many products using the same chromosome sequence. Now translation, we talked about transcription, which was making the RNA. Translation is a process where you convert that code on the RNA into making the language of amino acids. The ribosomes, which we compared it to the cook, that will start to read the recipes and start asking for the ingredients. Those ingredients will be carried by a transfer RNA that will bring to the ribosome the amino acids, and therefore the amino acid starts building. The very first amino acid on this chain will be methionine. That is the very first one, and on that you will start to build. Like I said, you will use the tRNA or the transfer RNA to fetch the ingredients that you need. That will bring in the amino acids. The amino acids will start to be organized and arranged next to the ribosome, giving you a polypeptide chain, and then the processing of that chain will give you the protein. Here is again the overview of the peptide formation. We start with the DNA, and here's a step that is not here, which is the making of the pre-mRNA. Then you have splicing. You get to the RNA, and finally you have this final mRNA that will be read by your ribosome, and calling in the carriers. Each of them, the tRNA will bring in one amino acid. You link the amino acids together, and now you have a peptide chain. That is not the final product. It has to be modified subsequently, and we will do so inside the endoplasmic reticulum, the rough endoplasmic reticulum, which will be allowed to be modified further. So the proteins are synthesized on the ribosomes. They will be inside the rough endoplasmic reticulum. They get more modification inside, and then they are sent to the Golgi complex or the Golgi apparatus, where it gets the final modification and processing and packaging to make those vesicles. Some of them will stay in the cell if needed, and some of them will exit out of the cells in a process we called exocytosis. Once again, here the proteins are assembled on the endoplasmic reticulum surface, and they are modified. This modification will be called post-translational modification, because this process happens, or the modification happened after the translation, after the amino acids were linked together, after the RNA was read. So we'll call that post-translational modification. This is actually very important. It will modify how long your protein will live and how functional is it, and the location of your protein will be determined by this, what we call post-translational modification. The vesicles will fuse, and they will unload into the Golgi cisterna. We did cover that, and the Golgi will pack, modify, makes the final modification, and now the proteins can leave the cell by a process we called exocytosis. Now, this is the part we start here with, the area that we did not necessarily cover in our lecture. We explained that the genes are not all activated at once. We have to maintain sort of a regulation of gene expression, and that regulation will determine when does insulin come. So insulin is not all the time on, and you have to think of your hormones, you have to think of your pumps, you have to think of your neurotransmitters. They are there when you need them, but they don't have to be all the time there. So you have to have some superior hand in dictating which gene is active and which one is not. And I compare that in the lecture to you with having desktops, 50 or so desktops, that have on their hard drives thousands of programs, but you initiate only 5 different programs on each computer, and now those computers are functioning differently. So although all of them have the same sort of code on their hard drives, but because you did not execute all the programs at once, then they will look and function differently. And the same thing with the cells. So we do have to maintain some sort of regulation element on the synthesis of our proteins, synthesis of our enzymes, and there are a lot of examples for that. For example, the testosterone production of insulin, We'll talk about all these different examples when we cover those chapters, but enough for you now to just realize and understand the fact that those genes are regulated. That is why women, for example, they only lactate when they are pregnant or shortly after delivery for a year or so. Why aren't they lactating all along? Because of the gene regulation, and we have to understand that you as a cell, you possess the ability to switch on and off genes as your needs come. So now we'll talk about a different aspect, which is the ability of the cell to divide. Just like, consider the cell like a corporation, and before you divide a corporation or a bank, you have to make everything exactly even. But if someone tells you, well, you can divide this branch into two, but make sure that the two emerging branches are of the same size, exactly the same size as the original branch. So what would you do? The first thing is you make your employees twice as much, and you make your computers twice as much, and then you make your walls and desks twice as much, and then you divide the two. Then each of them has equal number, and that equal number is the same number and the same the number of employees, computers and everything to that original one that you divided. And the same thing happens in the cell. So in order for you to divide the cell, you have to make sure that the coming cells, the cells that are resulting from that kind of division, contain the same amount of DNA, the same amount of mitochondria, the same amount of lysosomes, centrosomes, to that original cell. So the first step is to multiply everything. Everything else is easy. You can multiply mitochondria, you can multiply this, you can multiply that, because these are functions of the proteins. But how do you multiply the DNA? The DNA is the core. How do you multiply that? So we use you remember the DNA has two strands, and we use a law called a complementary base pairing, where we separate the two strands from each other of the DNA, and then you use each of the strand as a template to build on. And if we look at the picture, well, I don't have a picture here, but imagine it that you have a statue, and you can use each side to make a mold, and then now you have a complete statue, and so on and so on and so on. This is how we replicate the DNA. Why do we do it this way? Because we don't want any mistakes. We don't want each of your liver cells or your brain cells to contain different thing or look different than the original cell you started with. Or the blood cells. You want all the blood cells to be exactly the same shape and exactly the same DNA and exactly the same functions. Of course, things are not always perfect, and every now and then you have a mutation, which is a change in the genetic code, and that can lead to some serious consequences, and sometimes it can lead to cancer. So in DNA replication, you have an enzyme called the DNA helicase, which unzips the helix, this structure, the unique structure of the DNA, and that will initiate what we call replicate and fork. Essentially, the DNA strands are separated from each other. Now an enzyme comes called DNA polymerase, and they will read the exposed bases, and they will match them and build on top of them another mold of the other DNA strand, and now you replicated the whole DNA. So it's called semi-conservative replication, because each daughter DNA will contain one new helix that's synthesized from one strand from the original, and the other one is the mold that we just casted on top of it. And the process looks like this. Here you have the original DNA. Now you are separating, oh no, here is the original DNA. Now you're separating the two by an enzyme called the DNA helicase. That provides us with something called replication fork. Now comes an enzyme called DNA polymerase. It will start building on this strand, taking the free nucleotides and building on, and now we have the complementary strand to this one and the complementary strand to that one. If a problem happens, then we can cut and paste and cut and heal the problems that we are cutting with an enzyme called DNA ligase. You don't necessarily have to go into extreme details with that. New histones will be synthesized to complete the chromatin. Remember that the chromatin has both DNA and histones that the DNA is wrapped on. And in the end, now you have multiplied your chromatin twice. The chromatins will still keep attached to each other. The chromosomes that you just synthesized, they will remain attached to each other even as copies. We call those sister chromatids. So the DNA polymerase does not make mistakes, and if it does, it will correct itself. Just like a typewriter, you can go erase and retype the actual letter. Or there are checkpoints that will see, or checking enzymes, that will see that you actually wrote the right sequence, and if not, they will destroy that sequence and they will attempt to remake it, and if not, they will destroy that cells. And it's very, very important because we don't want too many errors, and because the more errors you have, the more chances for diseases like cancer to happen. With that, mutation, which is a change in the DNA structure due to replication error, sometimes due to environmental factors like radiation or chemicals, such as those emerging from diesel exhaust or from burning fossil fuel. Those chemicals have the ability to bind to the DNA and cause mutation. And if this mutation happens in a critical area, that can lead to cancer formation, of course. The cell cycle is the life cycle that extends from one division to the next, and there is we will divide that into what we call the G phase, or the gap phase, and that's in between the division. We have G1, and then we have S phase, and then we have G2, and then we have the mitotic phase. Let's start with the G1 phase. That's the first gap phase. It's an interval between the cell divisions, and it accumulates the material. Remember, when dividing the banks, you need to replicate everything, and you need to replicate the mitochondria, you need to replicate your ultrastructures, the organelles inside, and also you need to replicate your DNA. In the S phase, you will start to duplicate your centrioles, and you will finish replicating your DNA. In the G2 phase, which is the second gap phase, it's an interval between the DNA replication and the cell division itself. It finishes the centriole duplication, it synthesizes the enzymes, it repairs the DNA replication errors if they were there. And in the mitotic phase, which means cell division, or M phase, the cell will replicate the nucleus, it would pinches it into forming two daughter cells. The interphase is a collection of G1S and G2 phase. We'll call that interphase. The G0 phase are the cells that left the cycle for resting. So these are the cells that will never divide. Like the muscle cells, we know that the muscle cells, the adult muscle cells and the nerve cells will never divide again. So they become in what we call G0. Those are terminally differentiated cells that will never divide again, so we'll call that G0 cells. In mitosis, the idea of mitosis is producing two identical cells, so it will be involved in every division in our cell body except for gametes, which are our eggs and the sperms, eggs in the females and sperms in the male. The function of the mitosis is the development of individual forms of an individual from a fertilized egg, or growth of a tissue, or replacement of a tissue, or repair of that damaged tissue. So it is a dividing of the cell, and it depends why are you dividing the cells. Are you growing as an individual? Are you starting as a fertilized egg and then you are growing as an individual? Did you lose something? Is your liver damaged and now you need to repair that damage? Is your bone broken? Did you cut your hand? All that will involve mitosis, of course. The mitosis will involve four different phases, as as you learned in biology class, one we call prophase, second is metaphase, anaphase, and telophase. In the prophase, you will see the packaging occurs of your chromosomes into definitive structures. In the metaphase, your chromosomes will line up and your mitotic spindle will start forming in the metaphase. In the anaphase, the sister chromatids will be separated from each other by the pulling of this mitotic spindles. In the telophase, that is the actual separation of the two cells from each other. So let's look here. In the prophase, you are packaging the chromosomes into this structure and you duplicated your centromeres. In the metaphase, you're lining up the chromosomes, the duplicated chromosomes, in one line. We call that metaphyseal plate, and we have what we call spindles, mitotic spindles. And when you pull on those mitotic spindles, then you're separating the sister chromatids from each other, and now you have chromosomes on each side. That is in the anaphase, and in the telophase, you start to separate, you start to reform the nuclear membrane, and then you start to separate the two cells from each other. Then you will end with two cells that were identical to the one you started with. The telophase is the division of the cells from each other. The anaphase is the separation of the chromosomes from each other. The metaphase is when the chromosomes line in one horizontal line in the cell against, and then they are pulled into each pole of the cell by what we call centriole. And the prophase is when you are packaging the chromosomes. And as I always say, if you want to move to another place, if you want to move your home, you don't want to start dragging your tapes, your CDs, your cover sheets. The first thing in moving from one location to another is you package. So you put them in boxes, and this is the way the cells deal with this issue, by putting the DNA into those shapes of chromosomes, so it's easier then to divide the DNA. I always compare it. Imagine if two are living together and they had books on the shelves and clothes and stuff and all that they want to share, and each of them want to move separate from the other. What do? They package their belongings into boxes before they move, right? So the cells also will package their DNA into those chromosome structure before lining them up at the metaphase, and before separating them at the anaphase, and before the cell themselves actually separate from each other at the telophase. So we talked about those stages and the mitosis. One important area here, which is the mitotic spindle. It's very important, those cytoskeleton fibers that are pulling on the sister chromatids. The importance for them is in cancer treatment, because if you inhibit those mitotic spindles, then you disallow the cells to divide, and that is one of the aspects of chemotherapy, by inhibiting the mitotic spindles. We did cover the metaphase and the mitotic spindle, and we did cover the anaphase and the telophase already in the picture, as I earlier explained to you. In the final part, there is something called cytokinesis, which is the division of the cytoplasm now into daughter cells, and we can achieve that by a motor protein called myosin and microfilaments called actin, and those essentially will snap the two cells from each other. And I like to compare the cytokinesis by a process. If you have a large piece of dough, baking dough, in your hand, and you continue squeezing at the middle between the fingers or with a rubber band, and the two balls now of dough will snap from each other. And so it starts with what we call the cleavage furrow, and that will get tighter and tighter and tighter till the two snap from each other. Compare that to the dough, to your baking dough. The cells will divide when they have the stimulation by growth factor, when they also need adequate nutritional supply. Not all the cells will divide Some cells do not divide at all. We call that they enter the G0 phase. If they're snugly in contact with the neighboring cells, they don't have enough stimulation. They can also undergo inhibition of growth by the surrounding cells or by their own, telling them it's not time now to divide. Like our muscle cells and our nerve cells, they don't divide at all. Let's talk a little bit about the chromosomes and heredity. We did cover that in biology, and the heredity is the transmission of those genetic information that you are carrying on the chromosomes. Like how big is your nose, what color is your eye, how tall are you, how your skin looks like, how your eyes look like, how your eyebrows look like. All of these are traits that we have, but not all the traits are obvious. Some of them are inside us, like how much what kind of proteins are on the surface of your cells, and whether or not you have the gene that encodes for cancer, for example, a mutation of some sort, or you have a genetic disorder for a certain disease like achondroplasia. So all of these things can be carried by specific genes or alleles, and we'll talk about what are alleles in your body. So, but let's start with talking about the chromosomes. We have 23 pairs. One of those pairs will be called sex chromosomes, and 22 pairs are called autosomes or autosomal chromosomes. The 22 pairs are numbered from 1 to 22, and there is one that you inherited from your father and one you inherited from your mother. There is another pair that is the sex chromosome. You can have XX and you would be female, and you have XY and you would be male. That's the normal male and the normal female. Of course, there is some abnormalities, that you can have a female that has XXX, or you can have a male that has XY, or you can have a female that has X0. All these, of course, are chromosomal abnormalities that you can see. So here how they look. This is chromosome number 1, chromosome number 2, and as you see, although they are you have two pairs of them, they don't necessarily look exactly the same to each other, because simply you inherited this one from your father and this one from your mother. So they will look, the copies will look similar but not identical to each other. A diploid is any cell that has the 23 pairs. The haploid, these are only the gametes, like your sperms and your eggs. They will contain half as many only, so they will not contain 23 pairs, but they will only contain 23 chromosomes, and we call that the haploid. That's only present in the germ cells. If you mix two together, the genome from the sperm and that of the egg, you end up with a diploid, which is the fertilized embryo. There are certain terms that I would like you to remember. The locus is the location of a particular gene on a chromosome. The allele is different forms of genes at the same locus on two homologous chromosomes that will be called alleles. We have, since you have two different alleles, one of them you inherited from the father, one of them you inherited from the mother, sometimes one of them can dominate the other. And you did study that in biology when you compared the peas and the green peas and the yellow peas and the red flower and the white flower. We agreed into something called dominant allele that will present itself as a capital letter when you write that. Consider it that to be the stronger fellow, and if you have two people singing with each other, if both of them are high or strong, then both of them will sing equally. But if one of them is very dominant, you will hear that person much more, and the other one will be called recessive allele. However, if you have two people singing quietly, you will still hear both of them, because none of them is dominating over the other. So genes work the same way. You can have a dominant allele and you can have a recessive allele, and it's the combination between the two that determine your trait, or your phenotype. So let's say that you have a dominant allele here for healthy, and you have a recessive allele here for sick feature in your blood. If you have both your copies are the healthy ones, then you are disease-free. If you have one copy of the dominant and one copy of the recessive, you will be disease-free, but you will be a carrier. But if you have both copies to be a recessive allele that is diseased, then of course you can have that kind of disease. Those kind of traits or alleles can control your phenotypes, and here is an example of cleft chin. You can have either cleft chin or you unclefted chin. The cleft chin will be carried by the dominant allele, the dominant, which is the C capital. The genotype is the composition of the alleles, whether we have homozygous alleles, one dominant, one recessive, both of them dominants or both of them are recessive, or heterozygous alleles, you have one dominant and one recessive. They're not equally the same, and we call that heterozygous. But then the phenotype, so you have a genotype, which is the composition of the genes, and the phenotype is how observable the trait is. And if one allele is expressed over the other, then you will see the phenotype that corresponds to the dominant allele of that particular individual. It gets us to understanding what the genetic counselors do when they know that there is a certain problem in a family. Then they can advise that their kids may or may not have that disease, and they may suggest that they have to go through genetic testing when the woman is pregnant, because there is a high chance for the offspring to carry the same mutation that both of them are carrying, but in form of just an allele, and it's not demonstrating itself as a phenotype. But when the child has both bad copies, then that is a problem, of course, for that child. More definitions, we can have what we call gene pool, that is the collective genetic makeup of population, entire population, like the United States, or special ethnic group, Latin American or Asian, and we'll call that gene pool. If they are isolated from the rest of the world, like Scandinavia, certain areas in Scandinavia, or aboriginals in Australia, for example, they are breeding from within the group, and therefore they have gene pool that is unique from the other genomes of the populations in the world. Sometimes you have more than allele that covers the same trait, like the skin color. We have, even with the blood typing, we have 3 alleles that will determine the ABO blood types. In cystic fibrosis, we can have, which is a disease that can happen in the lung, and can result in the inability to get rid of the mucus secretion because it's very thick. Can have up to 100 alleles that can be responsible for cystic fibrosis. The alleles don't necessarily have to be one dominant and one recessive, but what you can also have is what we call codominance, is when both of them don't mind the existing of each other. Like you have 2 singers, one of them is bass and the other one is soprano. You can hear both of them, and that's fine. And in blood, we have that. The A and the B, they can exist as codominants, so you can have individuals with blood type AB that is a codominant example. In incomplete dominance, you mix the 2 together. So let's say you have white flower and a red flower, and if you breed those 2 plants, then you will get a plant with a pink flower, and that we call incomplete dominance. The incomplete dominance also dictates our skin color, although it's a little bit more complicated in human than the color of a rose or the color of a pea. But to some extent you get that if a person, the height of an individual sometimes is carried by what we call incomplete dominance as well, which it's a blending outcome. Not a single one can overcome the other allele, but together they blend and then they give you something in between, and we see that more in flowers and also our skin color and so on. So here is an example of what we call pleiotropy. When a gene is carried by more than one allele, you can decide, for example, the color of the eye, the color of our skin by more than one allele, and then you can have a multiple inheritance traits that can pass, like we have dark individuals, lighter individuals. It's not all black and white. We're not mice. Even mice have black, white, and gray mice. But think of us and think of all the population around you, how many colors and the talls and the heights of the individuals. You will see that we have many possibilities, and we call that pleiotropy. Some of the genes are carried on the X or the Y chromosomes, mainly the X chromosomes. We call those sex-linked diseases. These are very important, especially for male, because male only have one X chromosome, whereas female have two X chromosomes. And if one male is ending with a single copy of the X that is deformed or it has a problem in them, then although the mother is a carrier and it do not have the disease, and the father is free, but because you are inheriting the Y from the father and one of the X's from the mother, you can end up with actually having that disease. And we call those sex-linked genes or sex-linked alleles. And you can find that in your biology class, and cystic fibrosis is, for example, carried by one of those or actually muscle dystrophy, not cystic fibrosis. Muscle dystrophy is carried, and hemophilia is also carried by sex-linked gene. Now, two other aspects. The penetrance is the percentage of the population with a given genotype that will actually exhibit that phenotype. This is a definition for you for the future to know. It's not always just the DNA and the information that is carried will dictate how the gene will function, but also the environment surrounding you will dictate how the gene function, how much of amino acids you are taking, what kind of environmental exposure you will get, and whether or not that kind of environmental exposure, for example, can cause cancer. So there is a rule for the environment, there is a rule for even the normal functions of the environment, the color of your eye. You will need phenylalanine to make the melanin pigment, and without that you're not going to have much of the melanin pigment, and therefore you don't have brown eyes. But it's also the environment can precipitate a lot of problems and mutations that can lead to diseases and can lead to cancer. So that gets us to cancer. Benign tumor don't belong to cancer. Benign tumor will have a slow growth. It's a tumor, but it's not cancer. It's slow growth. It doesn't invade anything. It does not metastasize, and very easy to treat because you just remove it. The malignant tumor, on the other hand, it's fast growing and it spreads everywhere, and that is the danger because it starts destroying other tissues and sometimes critical tissues like brain or like the liver, or it can invade blood vessels, and it can also utilize all your energy and all your ATP, and leads to wasting, and leads to what we call cachexia, that can be debilitating, of course, because the person doesn't feel like eating, and at the same time your metabolic index is high, and you end up wasting, and we call that cachexia. The problem, of course, with malignant tumors, as I said, that they metastasize. They don't sit where they are, and not all the cancer cells will metastasize, but the more metastasis the cancer cells are able or capable of doing, then of course the worse this type of the tumor is. So therefore nowadays they test a lot this metastatic ability or capability of a cancer cell to determine how bad is this cancer. Oncology is a term that refers to medical specialty that deals with benign and malignant tumors. There's another term called tumor angiogenesis which is the blood formation that happens, or the regrowth of blood vessels, and that's stimulated by the tumors themselves. There are different types of tumors, and those you may or may not have studied in biology. Carcinoma happens in epithelial cells, lymphomas happens in lymph nodes, melanomas in the pigment epithelium, in the melanocytes, and we'll talk about that when we get to the skin. Leukemias are in blood-forming tissues, and sarcomas, not all the blood-forming tissues, one type of it, and we will get to that when we cover the blood physiology. And sarcoma, it happens in connective tissue, in the muscle, in the bone, any connective tissue, we'll call that sarcoma. A carcinogen is an environmental factor that can cause or facilitate the happening or the occurrence of cancers. Now, many of the carcinogens are known for us, like radiation, chemical, viruses. All of these viruses, like cervical cancer, is caused by papillomavirus, for example. That's why young girls are now being vaccinated against papillomavirus. But in addition, we have radiation. We know that that can cause cancer. Chemicals such as those cigarettes found in cigarettes, or in diesel exhaust, or industrial chemicals, pesticides. All of these guys are known to facilitate or cause cancer. We know that some tumors, some genes inside us are supposed to suppress the growth of the tumors, but we also know that the tumors oversmart us, and they silence those what we call tumor suppressor genes so that the tumor can grow bigger. Here's an example of Wells tumor. That's a malignant tumor of the kidney, and that occurs in children. It's just an example. You don't necessarily have to know these details about cancer. This is a physiology course, not necessarily an oncology course. The lethal effect can lead to cachexia, can invade tissues like the brain or the liver, and therefore those tissues will stop functioning.